webinar will be from Dr. Jan Forth, who for the past 5 years has been a postdoc at FLI in the German National Reference Laboratory for African Swine Fever Virus. His current research interests include African swine fever virus evolution, soft ticks as the arthropod vector, ASF genetics, and next generation sequencing and endogenous elements. Recently, Jan published a paper, a deep sequencing workflow for the fast and efficient generation of high quality African swine virus whole genome sequences in the journal Viruses, where they developed a workflow to get high quality next generation sequencing on African swine fever. Jan will now give his seminar an update on next generation sequencing.
Hey everybody, I hope you can hear me. Thanks for the invitation, happy to be here. To give you a quick overview over NGS and African swine fever, or mainly maybe whole genome sequencing might be a better term actually. So in the end, let's start with a historical view. So this is actually the number of ASF publications over the year. You see there are different events over time. So you see in the beginning, of course, there was the introduction into Southern Europe. Then down here, there was actually the first publication of African swine fever whole genome sequence. You see the eradication of ASF from Europe. And then you see now the new wave starting in 2007, and you see the number of ASF publications per year go up over time pretty quickly. When we now also enter the number of whole genome sequences over time, and this is the accumulated number. You see actually the first sequence, as I said, published in 1995. But since then, actually the number of whole genome sequences increased slowly, but recently quite quickly, let's say. At the moment, we have around 75 ASF whole genome sequences available from GenBank. Actually, I tried to count them yesterday. So this number is relatively new. So the question is, of course, why is this development? Why are the ASF whole genome sequences coming up in the later years more frequently? And of course, you all know the endemic or the epidemic situation of African swine fever, you know, the spread to Asia. So more people are actually affected by African swine fever. And the second thing is the costs have gone down in the last years very quickly for sequencing. Of course, this is for the human genome, but I think you can extrapolate that for sequencing in total. And you see the costs have decreased rapidly over the last years. So sequencing is, for ASF sequencing is getting more cheaper. And since more people are affected, of course, more people are doing it. So more sequences are coming up very quickly in the last years. So why is this, sequencing African swine fever still a challenge? When we look at the ASF genome, you know, of course, it is quite big. It has up to 193,000 base pairs and has a lot of genes, open reading frames on both sides of the genome in both directions. It is closed. It has inverted terminal repeats at the end. And it has some other special features. When we look here, this is actually our Illumina reads, MySeq read 300 base per paired end, mapped to an ASF reference, the Georgia 2007 reference. You see here in blue, this is the coverage over the entire genome. So this would be position 1. This would be position 193, 90,000 something. And you see everything where the line goes down, there seems to be a problem. And with ASF, the problem is that has a lot of some complicated motifs for sequencing actually. And one of the most complicated motifs, even for modern sequencing technologies, is actually homopolymer regions. Here you see the Georgia genome has actually regions where it has 17 C nucleotides in a row. And this is actually, as I said, even for modern sequencing techniques, very, very complicated to have a higher resolution in these areas. And these are the regions where you have problems in assembling a high quality genome, where you are sure that you have a correct or at least a near correct sequence. So let's have a look. How is the situation on ASF whole genome sequencing? Sequences available in 2011, actually, we had from 24 genotypes that we know of today, we had actually 9 available. And you see most of them actually were published around 2003. This was at this time the newest one, the Georgia sequence, which was done in Pirbright. And since then, actually something happened. Let's go through the years. In 2014, you see actually some new sequences came online, but you still see there's actually only one new genotype. Most of the new sequences that came up are on the known genotypes 2 and 1, which were, of course, the ones that were pandemic. In 2015, you see actually it's only a few additional sequences coming up. 2016, only one. 2018, coming up a little more. And now you see actually, one of the problems I will talk in a few seconds, but the genotype 2, of course, which is of broad interest because this is the one actually spreading in Eastern Europe and Asia. More and more sequences are coming up there. This is the situation 2019, actually some new strains, but again, mostly genotype 2 sequences coming up. This is the situation at the moment. You see again, a lot of genotype 1 and genotype 2 sequences, very few of the other exotic genotypes. So at the moment we have around 10, a little more genotypes from 24, but we are a bit far away from actually knowing most of the genotypes that are around. So what is the problem? The main problem is actually at the moment, we have a lot of genotype 2 sequences from Eastern Europe and Asia. They're only, over the last 10 years, they're actually more than 99% identical to each other. So they only differ in single nucleotides over the entire genome length. So we were not able to actually do some kind of molecular epidemiology. We do no idea on conclusions on origins of introductions. For most of this, very, very few variations we see, and we have no correlations on the gene or the protein function, or if there is some influence, because we don't know the function of most of the genes, as actually was told earlier this day. One good example is the ASF Belgium sequence. We actually did this and we had 15 single nucleotide differences to the Georgia reference, which is actually 10 years older. Quality control on the sequences. So we have with most of the, some of the sequences, I have to say, we have no metadata to validate if the sequence was done correctly or to reanalyze it to see if we see differences. And I mean, a single nucleotide difference is very, very easily mistaken, for example, by assembly mistakes or by mapping artifacts. So this is actually a big problem at the moment with the genotype 2 sequences. So we desperately need new strategies.
Jan, we can't hear you.
Can you hear me now?
Yep, that's better.
Okay, sorry. So you see on the left the phylogenetic tree, and you see on the right the map, and you see every color here is a dot on the map. And what you can see is that, okay, the genotypes are pretty well distributed over Africa, so we have some information. But when we now subtract all the sequences we have only from the domestic pig population, you see actually we know pretty, we have pretty few information on the actually sylvatic cycle. We have a few, but in the end, there's a sylvatic cycle all over Southern Africa. So there's actually information missing. So cooperation with researchers from Africa is actually very crucial here in this point to get together the resources, the strains that are available, and on the other hand, the technique, the methods, the people who can sequence, and also have the bioinformatics background and also the background in ASF sequencing and bioinformatics to get this sequences done. So what is actually now in progress at the moment? On the left side, you see a P72 phylogenetic tree. So these are actually all the genotypes that are around at the moment. And this is actually on the right what at least we have in progress at the moment. And you see we have in cooperation with Chris from Pirbright and also with Edgar Simulundo and Herman Chambara from Zambia, we have produced more whole genome sequences from different genotypes. We are still in the progress of validating some of them, adding more different sequencing approaches to validate the sequences, but we are pretty far along. So I hope we will be able to publish some of these sequences soon and actually expand the knowledge on different genotypes, on different setups of the ASF genome. And I can tell you there are main differences between the different genotypes. So what's also, of course, necessary and also very interesting is we need to identify interesting targets. So we need to go away from sequencing random strains of genotype 2, like say, okay, I have to sequence 20 isolates from Estonia or from Latvia or wherever, but we need to detect more specific, to pick more specific our targets for sequencing. Very good examples is here. We need to detect and analyze the rare variants that actually exist. For example, the Estonia variant, which was sequenced in 2014. It actually is a field virus, so it was isolated in the field, and it has a 14 kb deletion at the 5' end and some reorganization of 7 kb, and it has an attenuated phenotype. So this is actually something we can learn something from. We want to actually identify these. So what we need actually would be some kind of whole genome-based screening for ASF genotype 2. So have a method, a very easy method to just have to see if everything is there, is there some reorganization or something. And if we see something interesting, then we go a little deeper and say, okay, now we do deep sequencing. Now we look a little more specific into what's going on there, but without investing so much money and time in the first place to sequence strains that are almost identical. What we also need very urgently, and this is also something that Manuel said earlier, is of course, we know there is changes in cell culture adapted viruses. And we also have these, we've seen this in the past, sorry, on WSL cells, for example, there is adaptation. These viruses tend to lose genetic information, actually pretty extensive genetic information, around 20-25 kbs. So this is actually something we need to be aware of, and also we need to check regularly these isolates when we work on them on cell culture to see if something else happened, if we get off-target effects, or if we get something we didn't expect. Also very important, I think, is the part on resequencing old virus isolates. So sequences that are in a database for a long time. We actually had this with the Georgia 2007 sequence, which we resequenced and corrected a bunch of actually sequencing errors, which were introduced by an older sequencing method. And also database curation. This is very important point because the sequences are used by many people to produce to set up their experiments to build on their research. So they actually should be done with the most sophisticated methods we have to see that they are correct and that people are working with correct information. And of course, what we need, and this is actually very urgent, we need transcriptome sequencing. So we need to have updated annotations. We need to know what is going on at transcriptome level. What are the difference between different viruses? Because there's actually a gap in knowledge. And we have now some transcriptome analysis coming up from Pirbright. We have done some which might be published soon. So actually there is some information now, but we need more of that. Also very interesting topic is quasi-species variant analysis. There is not much known on ASF on that part. But from time to time you get some not evidence, but some information on there. There might be actually something going on, but we are not very far along there. So do we need more methods? Well, we have for example, this is only an example, the Illumina technology. We use mostly the MiSeq because we have it available, but most of the Illumina machines are very well suited for ASF sequencing. They have a high accuracy, they have a high output. You can do multiplex sequencing. Sorry. But of course, the read length is pretty short. You have problems with homopolymer errors, especially also with the Illumina platform, but especially iTorrent is very error prone in homopolymer regions. And of course, you have a long run and prep time, especially for the Illumina systems. In combination, of course, with other methods, for example, the Oxford Nanopore MinION, which is a third generation sequencing method. It produces ultra long single molecule reads. It has a very short runtime, a very short sample prep time, but of course some disadvantages. The accuracy is low, the output is low, and you need a high quality DNA sample actually. But in the end, put them together, and this is actually the point. So we need to combine the methods we have. We have enough good methods for sequencing, but we need to combine them. We need to be smart about that to get what we want in the end. And at the moment, we want high quality ASF whole genome sequences. So whatever, in the end, a huge problem, almost always the huge amount of host sequences in the background. So usually when we sequence specific let's say like tissues from infected swine, we are talking about 0.01 to 0.05% of viral reads per sample. This is actually, you have to do very large data sets to get enough reads to build your whole genome sequence. So, but also we have methods for that. For example, at the FLI, we use target enrichment. So you just capture out what you want to sequence, you clean it up, you amplify it, and then you sequence especially what you want to sequence, in this case, the ASF reads in your sample. Other people doing it the other way around to get rid of all the host sequences and sequence whatever is still is in the sample. This is also a good way. Both actually in the end bring you a removal of background sequences. You can do high coverage sequencing. You do not need to do as much reads, so you do not need to have much resources, computing power, and also money, of course. But of course, there's also some disadvantages. US amplification bias. It is pretty expensive, especially when you do the MyBaits capture or the target enrichment we do. And of course, with our methods, for example, you only capture known targets. So in the end, we have good sequences, we have good methods. The thing is, we want the quantity of the sequences. We want more sequences in the database, and there's where the quality part comes in. We need metadata and we need quality control on the sequences that people produce. So we need raw reads, we need coverage information, we need information about what tools we used, what platforms were used, and so on and so on. So the researchers actually can validate the sequence they find in the database and say, okay, I trust that sequence because I can see, okay, it was done from this and this. I can actually repeat the analysis if I want to. We have good methods, as I said, but we need to be smart about combining these methods actually to get, in the end, what we want, a good high quality sequences. And of course, what would be extremely helpful, especially in Eastern Europe and Asia, is the whole genome-based screening approach. So maybe, for example, combining Illumina with target enrichment with the MinION platform, for example, to fish out of a sample what you want to sequence, put it on a MinION in a very quick time, have a draft genome sequence where you see, okay, what is in and what is not. And of course, and this is also most important, I can suggest that enough is we need to have improved sample selection. So we need more genotypes. So we need to have cooperations with people in Africa, in Eastern Europe, wherever ASF is going around and something interesting's come up. We need to have more information on the Slovatic cycle, but probably is where all these genotypes are actually coming from. And of course, and this is, well, one point I have also to stress is we will, of course, do a whole genome sequence when we have a new introduction. So we, of course, want to know.
We lost you again, Jan.
Sorry, it's this button here. So in the end, we will still sequence new introductions into new countries, but we have to do that with a little more specific background. So with that, thank you for your attention. Thank you for the opportunity to say something about sequencing here. I'm happy to answer some questions. Thank you.
Thank you, Jan. Nice talk. I have a question for you. What do you think the value of genotyping as currently established in African swine fever?
You're referring to the P72 genotyping?
Yes.
Well, I have mixed feelings about that. On the one hand, of course, it's useful up to a specific end because you can have with a very simple tool have some kind of idea where a virus might belong to. But in the end, when you compare this with the whole genome information, you sometimes get a different picture. And this is actually what we have now, what we will see in the future with more genotypes coming up, that there are discrepancies between the whole genome information and the P72 information. So I think we will have to think about maybe new method or improved or modified method for genotyping in the future. But in a moment, I think it is how it is.
Next question is there evidence for recombination between genotypes in areas where several genotypes are present, such as Africa?
Actually, it is probable. There is some evidence that there might be actually most probably in the Sylvatic cycle there might be some recombination. It might be actually the tick that is involved here. I know from no reports from domestic pigs or from other swine that there is a recombination, but in the tick vector it might be actually possible. Yeah.
Next question is, Estonia 2014 seems to be significantly different from all other genotype 2 genomes. It was sequenced at FLI and published in 2018. Can you please elaborate on the rearrangements you observed there? Have you tried to resequence it to confirm these rearrangements and deletions?
I'm happy to. And yeah, we actually sequenced that in 2018, I think. It came from Estonia. It was a field virus which went here into animal trials. What we found is actually, it was also for us pretty surprising because we found a 14 kb deletion on the 5' end. But also we found a 7 kb duplication, so 7 kb from the 3' end were actually invertedly bound at the 5' end. So there was a deletion and there was a recombination at the same time. We sequenced that several times. We resequenced that. We did PCR on that. We sequenced the PCR amplicants because we also wanted to be sure that this was actually accurate. But in the end, yeah, we came up with every time the same thing. The recombination was there. And yeah, it's a very interesting virus.
Next question is, Estonia 2014 is attenuated. Have you done animal experiments?
Yeah, we have. Actually, it was published. Several experiments were published, also in the paper where the sequence was published, actually. So yeah, we have, and it seems to be attenuated to some point. At least more animals survived that with the wild type. Yeah, but in the end, and this is, of course, this is important, Sandra just intervened. It has not actually been, it is not in the field anymore. So actually it was probably overgrown by the wild type. So we don't know why for sure, but there are some speculations around. But in the end, it is a attenuated virus, so it probably does not kill as effective as the wild type. And since ASF needs to spread through blood and tissues of, for example, dead animals, also bleeding animals, probably it was just overrun by the wild type. But this is actually just a speculation. Yeah, but in the domestic pig, it would be not nice, yeah, because of the attenuated phenotype, of course.
All right, that looks like all the questions. Oh, no, one more. Besides Estonia and LV17, have you found more natural attenuated strains in Europe?
So far, I'm not aware of more, actually. We actually, at least, have not found more attenuated strains. I think these are the only ones. Yeah, exactly. 2, I think, yeah. No, I'm not aware of more.
You mentioned a simple technique to screen for genotype 2 viruses and make sure they differ from the ones we know before going for whole genome sequencing. Can you please elaborate on that screening technique?
Actually, at the moment, it's more a concept. It's not a specific technique, but at least it's the combination of two techniques. So we have, for example, the capture, so the target capture of specific sequences from a sample, and we have the MinION sequencing. One, actually, the target capture produces very pure samples of what you want to sequence, so the ASF sequences. And the other one, the MinION platform, produces a very simple and quick tool to get sequencing done. Of course, the MinION has a problem on its own. It's not very accurate. But in the end, if you have a high coverage, you can actually make up for the low efficiency of the sequencing. So in the end, the combination of both could be a valuable tool to get a whole genome screening approach. Another approach would be to combine, for example, the capture with Illumina iSeq technology, which is a very small one cartridge system where you could insert a lot of captured samples at once, reducing the price significantly for building one specific ASF genome. And using this just for a screening approach, and then in the end, going for deep sequencing on interesting samples. But this is, at the moment, it's more a concept.
Any advice to use MinION for ASF sequencing?
Well, depends on the question. If you want to do it for whole genome generation, usually you need high quality DNA. So prepare good DNA from your sample. If you want to deploy it in the field, I think this was actually a question that was raised earlier. Of course, it could be used in the field. The question is, it would have to be validated very good to see if the information you get from a field sample in the field actually can live up to the standards of a high quality whole genome sequence. But this in the end depends on the question you have. Do you want to have this from the field, or is it just you want to have some basic information in the field very quickly and then go to the lab and do the more sophisticated analysis there? That depends entirely on the question.
Do you know the background of the strains that was ever used as a vaccine in Spain and Portugal around last century, 1960s?
Not specifically, no. I have not looked on that into detail, no.
Have you tested to remove the host genome by using enrichment towards microbiome kits?
We have not so far tested that because the capture is working very well. I know another technique that was used to remove the host sequences. I think methylation was the key point here, but we have also not tried that because usually for us the capture is working pretty well.
Okay, I think that is all of the questions. With that, I think it's time to thank all the speakers. We had a lot of good talks today. And again, to thank the University of Pretoria for hosting the webinar. And we will see you guys hopefully all in Uganda in 2021.